In this section, we will discuss the osmosis and effects of ingesting fluids of differing osmotic potential. Osmosis is the passive movement of water across a semipermeable membrane, from regions of low solute concentration to those of higher solute concentration. As you can see, on right side, solvent has been moved from lower to high concentration of fluid. Biological membranes are semipermeable in that they usually allow the free movement of water, but restrict the movement of solutes. The creation of osmotic gradients is the primary method for the movement of water in biological systems. This is why the osmotic potential, or osmolality, of body fluids is closely regulated by a number of homeostatic mechanisms. A fluid at the same osmotic potential as plasma is said to be isotonic. But fluid at higher potential means more concentrated solutes is called as hypertonic, and fluid at lower potential is hypotonic. Osmolarity depends on the number of osmotically active particles, or molecules, per liter, irrespective of their identity. But osmolarity is number of active particles, or molecules, per kilogram of solvent. Hence, unit of osmolarity is osmol per liter, and unit of osmolality is osmol per kilogram. Osmolality is preferred by physiologists as it is independent of temperature, though in physiological fluids, the values are very similar. The osmolality of plasma is approximately 290 milliosmol per kilogram, mostly due to dissolved ions and small molecules. For example, glucose and urea. These diffuse easily across capillaries, and the crystalloid osmotic pressure they exert is therefore the same either side of the capillary wall. Proteins do not easily pass through capillary walls and are responsible for the oncotic or colloidal osmotic pressure. This is much smaller than crystalloid osmotic pressure, but is critical for fluid transfer across capillary walls because it differs between plasma and interstitial fluid. Oncotic pressure is expressed in terms of pressure, and in plasma is normally approximately 25 millimeters of mercury. Maintenance of plasma osmolality is vital for regulation of blood volume. Drinking fluids of differing osmotic potentials has distinct effects on the distribution of water between cells and extracellular fluid. Now let's discuss the effects of ingesting fluids of differing osmotic potential. In case of hypertonic fluid, plasma volume expands. Plasma becomes concentrated. Free movement of water and ions from plasma leads to expansion of interstitial fluid. Interstitial fluid becomes concentrated. Osmotic potential draws water out of cells. Intracellular fluid concentrated. As intracellular fluid is inside the cell, so what happened to cell? Well, cell shrink due to loss of water. In case of isotonic fluid, plasma volume expands. Free movement of water and ions from plasma leads to expansion of interstitial fluid. Fluid is isotonic, so no osmotic potential generated between interstitial fluid and cells. Intracellular fluid is unaffected, so cell remain unaffected. In case of hypotonic fluid, plasma is diluted. Oncotic pressure reduced. Water moves into interstitial fluid. Interstitial fluid diluted. Osmotic potential generated between interstitial fluid and cells. Intracellular fluid diluted. Cells swell as water moves into cells. Hyper means more. and tone means strength. In case of hypertonic solution, cell shrinks. Iso means same, and tone means strength. In case of isotonic solution, cell remains unaffected. Hypo means low, and tone means strength. In case of hypotonic solution, cell swells. This was all about today's video on body water compartments and physiological fluids. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.